This is a prerequisite so that you can attend other guys' lectures, like our own fellow lectures. So I'm doing this so that you can understand their classes better, especially people from high school backgrounds or undergrad backgrounds. Okay. So, introduction, what are biomolecules? A biomolecule, it's basically one of the many substances which are produced by cells or living organisms and are found basically throughout the biosphere, and they can have a variety of size, structure, and functions. So the biomolecule world can be classified as macro and micromolecules. So you guys get that?
Yes.
Okay, great. Because like you guys respond, otherwise I'm gonna think I'm lagging out. Yeah? Okay. So how are they classified? And is basically as if you classify them according to the solubility in water, it will be aqueous phase, which is true. Solution will be ions and micromolecules, basically like calcium, sodium, those ions. And colloidal state will be macromolecules the such as basic proteins and other protein-like substances. And then you have the non-aqueous phase. Okay, you have non-aqueous phase, which is the one which is not soluble in water. So, which is the structural components of cells, such as cell walls, the components of cell membrane, chromatin, etc. So, analyzing tissue, if before you analyze a tissue for its biomolecules it contains, you need to treat it with a specific procedure, which is basically you take a mortar and pestle and you grind it with trichloroacetic acid. It's another carboxylic acid with 3 substituted chlorine atoms. You grind it, then you take the thing you grinded and then you centrifuge it. It's centrifuge it with a high velocity, I mean high angular velocity, which is called ultracentrifugation, and then you get the soluble fraction, which is micromolecules, which is of mass less than 800 daltons, and then you have the acid insoluble fraction, which is more than 100 kilodaltons. If you don't know, dalton is another. It's another way of representing molecular mass. It is basically what I would call the opposite of a mole, or that's what. Okay, so these are the various micromolecules and macromolecules you'll find, which is amino acids, dipeptides, tripeptides. These are all basic proteins like amino acid chains. Then you find monosaccharides, disaccharides, trisaccharides, mononucleotides, and dinucleotides. They are the components of DNA, if you may know already, and these are sugars, or saccharides as you might call it. So macromolecules would be the larger components such as proteins, polypeptides. Basically, protein is a polypeptide, if I had to say. Enzymes, which are again proteins, polysaccharides, which are the polymers, and then polynucleotides, which is DNA, basically any polymer. And then we have one exception, which is lipids. You find them in acids insoluble fraction, but their mass is that of a micromolecule, but it's not a polymer. But so, can anyone explain why you cannot find it in the soluble fraction, but only in the insoluble? Anyone? Hello? Chip.
Yes.
So why is it so?
No, sir.
Oh, I think it's because
Yeah?
Because lipids are hydrophilic? Hydrophobic, I mean.
Yeah, they're hydrophobic, so they basically cannot dissolve in water because of their hydrophobic behavior. So yeah, good attempt. I like that. Damn, this is empty. Okay. Categorizing biomolecules, like in another way, primary metabolite, which is essential for metabolic activity of an organism, without which it cannot survive. Like if I take out all the carbohydrate, proteins, and fats out of your body, you'll be dead the next moment. It's like that. And then you have secondary metabolite, which is derived from the primary metabolites by some chemical procedures as byproducts, and they are not essential for the functioning of the organism, such as cellulose, rubber, gum, carotenoids, alkaloids. Alkaloids are, if you didn't know, morphine, codeine, cocaine, those types. So if you remove them from the organism, they don't give a damn, basically. Okay, next, so carbohydrates, hydrates of carbon, as the name says. Chemically, they are basically polyhydric alcohols, but they have a aldehyde or a keto functional group. If you didn't study organic chemistry before, it is aldehyde or ketone. You can easily recognize them by the double oxygen bond on the carbon atom. So basically, a carbohydrate with the aldehyde group is called aldose, while one with the keto group is called ketos. So carbohydrates in a living system exist in the form of monosaccharides, oligosaccharides, and polysaccharides. So monosaccharide is one unit, oligo is a few units, and poly is a lot of units. So, damn, this class actually be pensive right now. Anyway, monosaccharides, they're the simplest form of carbohydrates, the simplest. So one monomeric unit, and they have extensive power of isomerism, and then they can have a lot of isomers from their carbon configurations, and then the free aldehyde or keto group shows reducing property. Based on the number of carbon atoms, they can be trioses, basically 3 carbons. This is the most basic types of trioses or sugars. D-glyceraldehyde, so you have aldehyde here. This is the aldehyde group I was talking about. You see you have a oxygen atom on the carbon atom, and then you have a hydrogen. So that's the most basic part. And then you have 2 hydroxyl groups, which gives us the glycerol nomenclature. And if you're wondering what this G is, G is the optical configuration. It is dextrorotatory. So if you pass polarized light to it, light passing through one plane only, if you know what that means. So it will rotate the light to the right. So whatever initial direction it was, it will rotate to the right. And then you have this dihydroxyacetone. So the optical configuration is not given here. So you have pentoses. Tetroses also exist, but these are pentoses. And the basic pentoses are D-ribose, L-ribose, and ribulose. So you'll find them in the famous RNA molecule. Next, we have hexoses. The basic most hexoses would be D-glucose, L-glucose. So they are glucose. They have the aldehyde functional group, and they have a different optical property because of the configuration of the third atom. So because of the difference in their position, they have different rotating properties. This is the dextro version, dextrorotatory, and this is the L levorotatory. Okay, so, and this is the other keto isomer, because if you place a carbon oxygen on atom 1, you'll have aldehyde, and if you have it on carbon 2 or any other carbon, you'll have a ketone or a ketose here. So ketose would be fructose, especially in the fruits, and fruits, grape sugar, honey. Oh, grape sugar is glucose anyway. L-fructose, levulose. So this is their configuration. Again, on the third carbon, they have small configuration difference. Okay. So next we'll talk about the ring structures of monosaccharides. So we are using here something called Hayward's projection. So according to Hayward discrete projection, so sugars can form two types of cyclical structures: pyrene, 6-membered, which is this. It's a heterocyclic ring. As you can see, oxygen is inside the ring right now, and then furan. You won't find these double bonds in the sugar molecule, but this is a furan molecule. This is the pyrene molecule itself. Okay, so this is how they form the cyclical form. It's basically form a tautomerism. It bends, the molecule bends, and then it exchanges. The oxygen forms a bond with this carbon 1, and then the hydrogen comes over there, and then you have something called a cyclic monosaccharide. So cyclical form here is alpha glucose or alpha glucopyranose. Alpha glucopyranose, alpha D glucopyranose, because this is the dextrorotatory version, and alpha because of the geometrical isomerism shown on carbon 1. So since this is below the plane, it's alpha. If it was above, it would be beta, because after alpha comes beta. So this is my explanation of alpha nomenclature in short, because I've had to remove alpha nomenclature from this lecture because we wouldn't fit in the time limit. So after that, the cyclic ring provides a more stable configuration, and compared to the linear form, the Fisher projection. So these are a few examples. Alpha D fructofuranose. So can you identify which molecule was it at one point of time, the linear form? And anyone? Yes. Yeah, if you cannot speak, you can write in the lectures chat. I can see it. Okay. So basically, fructofuranose was fructose, D-fructose, because you have 1, 2, 3, 4, 5, 6 carbons here. And then you have ribofuranose, which is basically 5 carbons. Okay. Anyway, Derived sugars, because you cannot do work with one molecule on your own. So you have these 4 types, basic types. Deoxy sugar, which is, you have the hydroxyl part which is removed basically, and then you have a bare end, deoxy sugar. So this here is deoxy deoxy wait, can you guys guess it? Okay, this is ribose.
গরম গরম কচুরি আছে কে ওইটা
Okay, so this is ribose, and sorry, I got interrupted. And then you have amino sugar, which is the OH on the carbon 2 position. This was the carbon 2 position, which is being replaced by an amino group, which is right here, amino group. And then you have a sugar acid, which is, if you take a linear chain and then you add a carboxyl group at the other end of the molecule, you have a sugar acid. And then in a sugar alcohol, you basically replace the aldehyde group by an alcoholic group. So next, proceeding, we come to the next section, oligosaccharides, which is oligosaccharide by definition is a polymer of 2 to 9 monosaccharides linked by glycosidic bonds. So a glycosidic bond is a dehydration linkage. Basically, if you do know organic chemistry a bit, it's a dehydration linkage. That's what you call it. And in a dehydration linkage, you take the OH of one carbon and never mind the term anomeric. We'll explain in next lecture. So this one carbon reacts with any other hydroxyl group of another carbon, and they react. Anyway, they react and they release one water molecule from it. So by nomenclature, you'd have disaccharides, trisaccharides, and tetrasaccharides. These are the most common 2 examples, maltose and sucrose. And then you have some few rare molecules like raffinose and stachyose. So if you do want to know more about them, you can search about them. That's actually your lecture homework. So you have let's discuss about a few disaccharides. You have maltose. It's a basic disaccharide of 2 glucose molecules. You have one here, and then you join them by a 1,4 linkage, 1,4 glycosidic bond. You have carbon 1, then you have carbon 4, and then you join them. Then you have an isomer of isomaltose, which is an isomer of maltose itself. And in that, you would link carbon 1 with carbon 6, like this. You join them. So that's an isomer of maltose. If you don't know what an isomer is, it is a molecule with the same chemical formula but another type of configuration of carbon atoms, like the best I can explain right now. Then you have sucrose, another basic molecule. You have, instead of two glucose, you have a glucose and a fructose. So you have a combination of these two molecules linked by an alpha 1 beta 1 linkage. If you do know what alpha and beta are, or if you learned about it before, so alpha glucose, it was basically formed by an alpha glucose and a beta fructose. So as you see, the reducing ends, they get linked together. The aldehyde groups, they get linked together, and then they lose their reducing ability and they become non-reducing by nature. So next section we have polysaccharides. Polysaccharides means 10 or more units. You have oligo 2 to 9, mono 1, and then you have 10 or more. So for classification of polysaccharides, you have homopolysaccharides and heteropolysaccharides. So homo means they say monomeric units, and hetero means you can have more than one monomeric units forming the structural chain of the polysaccharide. And here are a few examples. Pentosane and hexosane are two subtypes of homopolysaccharides. You have pentosane, which means 5C, and then you have hexosane, which means 6C monomers. Let's talk about one of the most common polysaccharides you might have heard of. So starch. Starch is basically a polymer. So polymer means a lot of molecules linked together of alpha D glucose, alpha dextroglucose, or alpha dextrose. Starch has two types of chains: amylose, which is 1,4 linkage, and then you have amylopectin, 1,6 linkage, 1,6, and then you have 1,4. So that's that. So the linear chains are called amylose. These are branched, and linear chains are 20%, and the branch chains are 80%. So if you didn't know, when we test iodine, when we test iodine for starch, so if we add iodine and then we see blue color, so that blue color is due to the amylose. The amylose, they trap the iodine molecules and they become blue. While amylopectin, it traps some more iodine molecules and then becomes red in color. So blue, red. So this is the most abundant organic molecule in the biosphere. So it's much more abundant than any other polysaccharide you might find. So it's basically it's composed of very long chains of beta D glucose units, and these chains are weakly bonded by hydrogen bonds. If you didn't know what hydrogen bonds are, so it's basically between the hydrogen of one molecule and then the oxygen, in this case, of another molecule. So it's kind of like a weak force of attraction, like van der Waals forces you might have studied before. So it can contain from 800 to up even more than 15,000 beta glucose units, and sadly they cannot be digested by humans. Can anyone tell me why? Oh, is everyone asleep?
Mm-hmm.
Okay.
What do you mean?
Yeah, basically, yeah, you're correct, Hannah. We don't have the enzyme for it. Cows and other herbivorous animals have the enzyme for it. You might know it as cellulase. It has the ability to digest the beta 1,4 bond, break it down into glucose. if you didn't know, a fun fact, if we did have cellulase, we wouldn't have to suffer from hunger, and world hunger actually wouldn't be a thing. So that's a fact. Okay, so this is the next type, chitin, second most abundant organic substance after cellulose. So the main difference between starch and the chitin is amine group. Amine group right here, the acetyl amine group. Amine is because of the N group, and acetyl because of this CCCO, this thing. So it's basically joined at the position 2 of this glucose molecule. So chemically it is called N-acetyl because if you take the acetyl and it is attached to the nitrogen atom, you call it the N-acetyl, N, because it is linked to the nitrogen. And then you have the beta version of glucose, which is N-acetyl glucosamine. So they are linked by 1,4 bonds, again like cellulose, and it's a polymer. You can have N repeating units. And then you have a few other examples of polysaccharides, which is agar. It's used a lot in molecular biology experiments, PCR, and other bacteria culturing experiments. It is a mucor polysaccharide from red algae, mucor because it is rather sticky or slimy. It is a polymer of agarose. Agarose itself is another polymer of galactose. It's a homopolymer. Then you have agaropectin, polymer of glucuronic acid and pyruvic acid. As you know, pyruvic acid is also found in cellular respiration. So it's a fact of biology, biochemistry, that one molecule can be used in more than one processes. You can find it in more than one place. And then you have peptidoglycan. Oh, I didn't finish it here. It's another slimy mucor polysaccharide you find in bacterial cell wall, which is again a polymer of N-acetyl glucosamine and N-acetyl muramic acid. So it's another heteropolymer. So you find this in bacterial cell wall. It gives the cell wall some weird properties which can give it the potential to cause infections, pathogenicity, and that's it, I guess. That's a Biomolecules 1 lecture right here. And next session, we'll talk about glucose fructose again, and we'll talk about the optical isomerism, and then we'll talk about fats, proteins, and then vitamins and minerals also as well. So take care, good night, and I'll see you later. Bruh. So yeah, lecture is done.